As we just learned, there are three different types of lymphocytes that we are going to be concerned about: B cells, helper T cells, and cytotoxic T cells. And remember, one of the features of immunology is this concept of specificity. That is, there are specific T cells and B cells that are designed to recognize particular antigens. As we mentioned earlier, within the B cells and T cells, there are proteins referred to as antigen receptors. These are the molecules that are capable of recognizing specific antigens. So let's suppose that you're exposed to a specific antigen. It gets inside of your body. Whenever a B cell or T cell that has the appropriate antigen receptor recognizes that particular antigen that has just entered your body, then that B cell and T cell will proliferate. That means that it'll divide over and over again. And also it differentiates. And what this simply means is that two different kinds of cells will be generated whenever the lymphocyte divides over and over again. So clonal selection is this process, which we will see in the next picture, of a lymphocyte proliferating, dividing repeatedly, and differentiating, becoming different kinds of cells. So the end result of clonal selection is a clone. A clone is a population of identical cells, all recognizing the same antigen as the original cell from which the clone was derived. So in other words, whenever an antigen enters your body, there will be just a few of the particular lymphocytes that are able to recognize that particular antigen. But after the lymphocytes undergoes clonal selection and divides over and over again, then what will happen is you'll have a large number of cells of the same type that are able to recognize that same antigen. This process of clonal selection occurs in secondary lymphatic organs and tissues. This includes places like your tonsils and your lymph nodes. So whenever a lymphocyte undergoes clonal selection, it will produce two kinds of cells: effector cells, and this includes active helper T cells, active cytotoxic T cells, and a plasma cell. And these are the cells that will carry out the particular action of the immune response, but they will die afterwards. Plasma cells are the name of the cells that are derived from the B cells. The other type of cells that are produced from clonal selection are called memory cells. These cells don't participate in the initial immune response, but rather what they'll do is they'll respond during the second and subsequent invasions of that same antigen. And when these memory cells respond, they proliferate, divide over and over again, and differentiate in order to make more effector cells and more memory cells. And they have long lifespans. And the purpose of these memory cells is to give you the ability to very quickly fight off that same antigen if you're exposed a second or subsequent time. In fact, what they do is they respond so quickly that you don't get any symptoms from that second exposure, and you don't get the disease because your body fights off that antigen very quickly. Here we can see a picture that illustrates again clonal selection. As you can see, whenever a cytotoxic T cell, helper T cell, and B cell are activated, that is, when they're exposed to a particular antigen, they will divide over and over again and make active cytotoxic T cells, memory cytotoxic T cells. There will be active helper T cells, memory helper T cells, and plasma cells, and memory B cells. It's the active cytotoxic T cells that are going to do the actual action of killing the antigen. Plasma cells, what they do is they secrete antibodies which will bind to and inactivate the antigens. And again, the memory cells of all three types stay in the body and simply are able to respond much quicker if you get exposed a second time to that same antigen.